Hello everyone, today we are going to go over Mitosis, chapter 19, and this is actually the first lecture I've had to re-record this semester. So you may have noticed some of the packets are slightly different from what yours look like, but I check them all before and make sure it gives you at least the information that you need. But this one was a little bit too different, so I'm going to re-record it. And also, you've probably noticed that the page numbers on the previous lectures don't match up with what your packets might say because we got a new textbook this year. But hopefully that didn't confuse you. If you're ever confused, send me a message on Canvas and I'll help you out. But today we're going to look at what is a chromosome and what is it made of, compare and contrast autosomes and sex chromosomes. What does it mean to say that human body cells are diploid? Compare and contrast chromatid, sister chromatids, and homologous chromosomes, so some vocabulary there. What is a centromere and what is its function? Outline the events of the cell cycle, and also the 5 phases of mitosis and cytokinesis. And our question of the day, which remember shows up as extra credit on the exams, are how do cells check themselves before they wreck themselves? And I should have looked up who this was beforehand. I'm feeling super old, but I'm assuming he said something along the lines of check yourself before you wreck yourself. So we'll get to that at the end. And here's your pre-lecture assignment. This should already be filled in, but if you have any questions on this, let me know. And let's get into it. So during cell division, we're going to do mitosis first, which is for asexual reproduction and repairing and replicating cells, and then we'll do meiosis, which is involved in sexual reproduction. So the cell that divides is called the parent cell. And the new cells are called the daughter cells. And each of these daughter cells are genetically identical to the parent cell, disregarding mutations. So we talk a lot about chromosomes. So what is a chromosome? You can see them pictured down here. That's what we're referring to. In a resting cell, DNA is unwound in the nucleus. So you kind of will just have the DNA in the nucleus just strewn about, kind of like a tangled ball of yarn, and then little sections of that are genes that code for specific proteins. And so it needs to be kind of unwound, so as the cell is doing its everyday business, that it can find those genes and make the proteins that is needed. But in a dividing cell, the protein needs to be packaged so we can evenly divide it between the two new daughter cells. So what a chromosome is packaged DNA. So kind of what it looks like is DNA is wound around a protein called a histone. And it shows it here in this picture. These purple structures are the histones. This is the DNA, and it kind of wraps around those, and then wraps and wraps and wraps some more until you end with a chromosome. So kind of think of this as the histone, and the DNA wraps around it. Another histone nearby, it starts to wrap around it just to kind of organize itself into a chromosome. So the histones and the DNA together we call chromatin. So in our picture here, as it starts to wrap around the histones, we are now referring to it as chromatin. And keep in mind that one more thing, each human cell, and you've got trillions of them, has about 6 feet of DNA inside each of those tiny cells. Fun fact. Human, if I could spell. And something important to remember is that the number of chromosomes does not determine the complexity of an organism. So if you look at this table over here, we've got a human. We have 46 chromosomes. Dogs have 78, pufferfish 42, a cat has 38, pea plant has 14, sunflower has 34. There's another one, I forget what it is, but it's some seemingly insignificant organism that has a very high number of chromosomes. So just keep in mind it does not determine the complexity. So again, in humans, every cell in the human body has 46 chromosomes. Half of these, so 23, are from mom. And the other half are from dad. Which is why you look kind of like your mom and kind of like your dad. You're getting genes and traits from each of them. So here we have a human karyotype. That's a picture of all the chromosomes. And they're just assigned a number based on length. So 1 is the longest and 22 would be the smallest. So 22 pairs are autosomes. So they do not determine biological gender. But they code for pretty much everything else. So in this model here, all of these, with the excluding these two, are autosomal chromosomes. You have one pair of sex chromosomes. And those determine biological gender. So on this karyotype here, it can be XX, which codes for a biological female, and there can be XY, which codes for a biological male. There are some exceptions to this. For example, because of non-disjunction, which I think we talk about in mitosis, someone could be XXX, XXX, or they could be XXY, I believe, and that will result in some different disorders, but we're not going to get into those today. On to the next page, diploid versus haploid. So think of ploid as one chromosome set. So humans have 23 sets or pairs of chromosomes. And anytime you see di, that means 2. So we call it 2N, basically 2 sets of chromosomes. So we have 2 sets of chromosomes in the nucleus. And like we mentioned earlier, one from mom, one from dad. Haploid is just one set of chromosomes in the nucleus. And an example of where you will find this in humans are in eggs and sperm cells. And those are sometimes referred to as gametes, if you hear that word. And different organisms have different diploid and haploid numbers. So on this chart again here, you can see diploid is always double what the haploid number is. Just divide the diploid by 2 and you get the haploid number. So chromosome nomenclature, or what do we call these parts? So a chromatid is a single unreplicated chromosome. So here we have a single chromatid. It's not paired up with anything like you see down here. Sister chromatids are replicated chromosomes. So here we have our replicated chromosomes. They're sister chromatids, and they should be genetically identical. And this structure right here is called a centrosome. Or sorry, centromere. Connected by a protein called a centromere. And that's going to act as the attachment point for the mitotic spindle when it's time to do cell division. So the mitotic spindle are microtubules that move and pull apart chromosomes when it's time to divide the cell. So this is teeny, teeny, tiny. We've got centrioles here. These stringy looking things are the microtubules. These are the chromosomes lined up along the center. This is actually metaphase, which we'll get to in a minute. And then those microtubules will attach to that centromere here so they can pull the sister chromatids apart. Homologous chromosomes, if you see homo, that means same. So they have the same chromosome number in the karyotype. And again, you get one from mom, one from dad. So in a non-replicated chromosome, we would have these single chromatids. One is from mom, one is from dad. But here we have this replicated DNA, homologous chromosomes, and one is just the replicated version of what you got from mom, and the replicated version of what you got from dad. All right, let's get into the cell cycle. So actively dividing cells go through the cell cycle. And here's our diagram of the cell cycle over here. So you can see all of this in purple here is what we call interphase. Inter means between, so it's kind of like when they're not necessarily actively dividing. And 95% of a cell's time is spent in interphase. During interphase, the cell is preparing for cell division, and it can be further broken down into 3 phases. So you've got the G1 phase, which is when the cell grows and replicates organelles. So I think G1, G for grow. So that's this little section here. The S phase in the teal there is when the DNA replicates. You gotta share 2 identical sets of DNA between these 2 daughter cells, so the DNA is going to replicate itself in this S phase of interphase. And then in the G2 phase, the cell grows and makes proteins for cell division, like microtubules, so they can help separate the DNA. Remember, those microtubules are the proteins that are going to grab onto the centromeres to help separate those sister chromatids. So now we're into mitosis. This is when the nucleus and the DNA of the cell actually divides. The nucleus of the cell divides in two, and the chromosomes are divided, and it's comprised of 5 phases. Got prophase, prometaphase, anaphase, and telophase. And we'll go into a little more detail on those in a second. But you can see here, this is just the actual mitosis is only about 5% of the cell cycle. I'm sorry, this is small. I don't have my larger diagrams printed off today. But we've got those stages here: prophase, prometaphase, metaphase, anaphase, telophase. And then next comes cytokinesis, which is when the rest of the cell, besides the DNA, divides. So mitosis is for dividing the DNA, the genetic material, and cytokinesis is for dividing everything else, because those new cells need all the organelles, cytoplasm. So that brings us to our next point, is cytoplasm is divided between the 2 cells. And this includes organelles and cytoskeleton, etc. Alright, so an overview of what mitosis is for. It's a form of sexual reproduction. Asexual reproduction. So some organisms can do sexual reproduction and asexual reproduction, and some just reproduce asexually. And this is human biology, so I'm not going to get too much into it, but things like single-celled bacteria reproduce with mitosis or asexual reproduction. And like I mentioned before, this creates 2 genetically identical daughter cells. So here we have this picture again. We have our parent cell. This is the DNA inside. Through those phases of mitosis, the DNA is copied. Actually, sorry, that's an interphase. And then we go through the phases of mitosis and we end up with 2 identical daughter cells after the cell fully divides in cytokinesis. The daughter cells are diploid, meaning that they still have 2 sets of chromosomes each. And in humans, what it's used for is growth of certain tissues, repair, and to replace dead cells. Now to get into a little more detail on the phases. Sorry, I'm kind of losing my voice a little bit. When I was in college, we just learned the mnemonic, is that what you call it, as PMAT. But we've added this extra phase here since, and so now it's PPMAT, which reminds me of something that a brand new puppy would pee on. Rover, make sure to pee on your PP mat. I don't know, maybe that helps you remember. But step 1 is prophase. So pro kind of means before, so this is kind of before all the stuff goes down. And the nuclear envelope dissolves. So we've got the nucleus here with all the DNA inside, and if we're going to split that up, we need to dissolve this membrane surrounding the nucleus so we can split it up. That chromatin, which is just the messy ball of yarn inside the nucleus, is going to condense into those chromosome packages so we can easily split them between cells. You can see here we're starting to condense this mess into chromosomes. And these mitotic spindles form. So that's prophase. It's still considered a part of mitosis, but it's just kind of where everything's getting ready to be divided. Prometaphase, one easy step. Mitotic spindles attached to the centromeres. Which are these little structures here that connect the sister chromatids. And those mitotic spindles are going to start to attach to those so they can be pulled apart. Metaphase, I remember M, middle. And this is where the chromosomes align at the cell's equator or in the middle of the cell. So if you ever see a diagram here, if they're lined up along the middle, you're most likely looking at metaphase. And here these mitotic spindle fibers move the chromosomes. And the fourth step is anaphase, and how I remember if I see a diagram is these kind of look like little shapes as they're being pulled apart that look like capital A's. So this is when those sister chromatids are pulled apart by those attached spindle fibers. Sorry, got people coming in and out here. Okay, the last phase of mitosis is telophase. And as you see in the picture, this is when the chromosomes arrive at opposite poles or sides of the cell. the chromosomes start to unwind. Back into their tangled yarn mess. The nuclear envelope starts to reform. And the mitotic spindle reforms. Basically, we're going back to business as usual. So that's the end of mitosis, or the end of dividing the DNA. But we still have to split the rest of this up here, and that is cytokinesis. That's when the cytoplasm is divided among the two cells. So that's the rest of everything else. It's not pictured here, but all the organelles, the cytoplasm. So now we have two brand new daughter cells. And it's not really showing it here, but this will kind of come together and pinch off to split them apart. Cells pinch off into two daughter cells. And there you have it. That's mitosis and cytokinesis. That's cell division for asexual reproduction, growth, repair, what was the other one? Growth, repair, and replacement of cells in the human body. And our question of the day: how do cells check themselves before they wreck themselves? So this brings us back to the cell cycle, and there are 3 checkpoints in the cycle. The cell will make sure a cell is okay to move on to the next part of the cycle. If everything is not going perfectly here in the cell cycle, something's wrong with the cell, maybe a mutation, the DNA didn't copy correctly, the cell's going to want to stop that because we don't want an abundance of abnormal cells that just continue to divide. So this happens in 3 different parts of the cell cycle, and the first one is G1. So right here, in interphase, where the cell just kind of decides, hey, am I okay to divide? Is the cell okay to divide? Over here in G2, did my DNA replicate correctly? We don't want a bunch of mutations, so the DNA replicated here, and as it's preparing for mitosis, we got to make sure, did that DNA replicate correctly? And the third one is in metaphase. When they're lined up along the middle, are my chromosomes attached to the spindle? Because if not, they may not pull apart correctly, in anaphase, and you'll get an uneven number of chromosomes in each of the cells, which is not what we want. And that is it. As always, message me with questions, and I hope you guys have a great day, night, evening, whatever it is for you. And I can't figure out how to pause this.